All right, we're going to go ahead and get started with our final session of the day. All right, so I'm Alston Ham from East Tennessee Children's Hospital, and I want to commend everyone on their dedication in reaching our final session. And, it gives me a lot of pleasure to introduce Dr. Paldeep Atwal. Dr. Atwal's primary medical training occurred in the United Kingdom. He then completed genetics residency at Stanford University, followed by a fellowship in biochemical genetics at Baylor. He now serves as an assistant professor of medicine and medical genetics with the Mayo Clinic, and he is the medical director for the Center of Individualized Medicine here in Jacksonville. And he's going to talk to us a lot about his clinical experience with the Individualized Medicine Center, and then also a little bit about metabolomics.
Is this on? Okay, it's on. So first question people ask is they didn't realize there was a Mayo Clinic in Jacksonville. It's about 10 minutes up the road. And so I have no disclosures. And so my talk is going to be split into three quite distinct talks. One is talking about the Department of Clinical Genomics, Center for Individualized Medicine, and the Mayo Clinic Biobank. There's been a lot of interest recently with the $142 million NIH grant that will serve as the storage center for the Precision Medicine Biobank. I'm going to do a quick clinical case review of some interesting cases we've seen. And then I was going to talk extensively about metabolomic profiling, but you've already had that talk from my colleague at Baylor, Dr. Elsia. So I'll just give one or two quick clinical examples that she didn't talk about. So I won't go too much over this. Basically, sequencing is much cheaper than it used to be. The only thing I'd highlight, though, is the understanding of genetic knowledge and the number of validated single base variants in the human genome is increasing, but not at the rate as the cost is decreasing. So while knowledge is increasing, it's not at the same rate as the technology. And then everyone is, in the general press and the population, very interested in genetics and what it means for certain diseases, carrier testing, genetics of common disease, obesity, cancer, Alzheimer's, and even things like human longevity. So there's lots of interest out there for genetics. And people are, the goal in all of this kind of personalized, individualized medicine era is to move from the traditional model of medicine, the anatomical model, to the genomic model. So Mayo has decided that they're going to do their best to be one of the leaders in this field. So, and around 2007, the Center for Individualized Medicine was formed with a number of translational programs. The main one, and the one I'm mostly involved in, is the clinical program or clinomics program. But there are others that I won't talk about today. The microbiome program, looking at how the microbiome affects our health and disease. Pharmacogenetics, looking at how our bodies metabolize different drugs. And there are large clinical trials going on in that, such as the trial looking at clopidogrel or Plavix and the pharmacogenetics for that and how that influences people post-MI and their outcomes. Epigenetics, one way to control gene expressions, and then biomarker discovery is another one. So the infrastructure support programs are also put in place. I'm going to talk a little bit about the biobank in a minute, but there's also a group of biomedical ethicists, bioinformatics, education. There's online courses for residents, fellows, and other health professionals, and also there's a annual conference in October that these days gets around 1,000 people attending. And that's in Rochester, Minnesota. Obviously IT support, and obviously the medical genome facility where the sequencing actually takes place. So a little bit more about the biorepository and the biobank. The The infrastructure is to establish and manage a state-of-the-art facility, and then there's to store the samples and distribute, etc. And then there's part of that is the collections, how to manage the collections through the biospecimens accession program. The main site is in Rochester, Minnesota, with a smaller site here in Jacksonville, Florida. The main site has a huge 35 square foot complex with these amazing robotic freezers, kind of space age stuff. This Hamilton BIOS machine has a 3.5 million tube capacity, and there's room to get 3 more of them if needed. And there's a robust disaster plan and all sorts of things like that. So the initial biobank that Mayo started independently was to enroll 50,000 Mayo patients, regardless of health history. So any patient that comes in, they get a flyer, are you interested in enrolling in the biobank? And it was there to provide an institutional resource for research, clinical studies. And it was done in a way to not need reconsenting and not need to start again for every researcher if they when they wanted to start a study. So the consent form allows for the specimens and data to be used in unlimited studies, past and future access to their medical record, future contact is allowed up to two times a year, and there's a certificate of confidentiality as well. Multiple different blood samples, 3 10 ml tubes for various different things you can do with that. And then so far, there has been there's around 56,000 people that have had their samples stored in the biobank. We've sequenced around the first 1,000, and there's plans to sequence eventually all of them with other omics platforms like Metabolomics has been talked about and looked at for some of those samples, and potentially all of them at some point down the future, although funding for that has not been secured. In terms of where they come from, as you expect, mostly based in the Mayo sites, heavily concentrated in Rochester, Minnesota, with some people in Florida and Texas as well. I can't really explain why there's so many from Texas, but there you go. The length of the electronic health record that goes with them, on average, is around 14 to 15 years. So on average, any sample in the biobank, you can access a fully complete health record that spans around 15 years, which is pretty amazing. And it's actively searchable. I've searched it. You can have queries and that kind of thing that they can do for you. And this is open to any researcher, inside or outside Mayo. Okay, so the next part I'm going to quickly talk about is the clinical services that the Center for Individualized Medicine offer. So the main things really are cancer, pharmacogenomics, and diagnostic odyssey. My 99% of what I do is in the diagnostic odyssey, but I'll give you 1 or 2 examples of the other things. In terms of how effective they are, it's not really any different from what anyone else is reporting. In terms of the diagnostic odyssey, 20, 25, 30% success rate in exome sequencing. And then the cancer, doing tumor sequencing, you'll find a sequence-based druggable target around 50% of the time. Of course, that doesn't mean you actually get a drug to give that patient. There's issues of, well, there's many issues, insurance, timing, and access, and all of those things. So even though you can find something that's potentially druggable, it doesn't mean you're actually going to be able to use that information for the patient's benefit. Patient example, the kind of stuff that's going on in the oncology, genetics type area. So 50-year-old patient, advanced cholangiocarcinoma, cancer of the bile ducts. has had the usual first-line treatment and not responded. Now bedbound, limited nutritional intake, really going downhill. So this patient was enrolled in whole genome and transcriptome sequencing, a research protocol, right at the start when they presented, so not after everything's failed, which would be too late, but basically the sample is taken right at the start during the first line of treatment, so the results are available at the time of refractory disease. And so there was a tyrosine kinase inhibitor that this patient could potentially have benefited. So they gave him that drug, and you can see the mets in the liver have all but disappeared. So that's a clear success story that is, not the most common story, but it shows the clinical utility of these methods. Pharmacogenomics. So pharmacogenomics is looking at how our bodies, the genetics and the enzymatic processes that metabolize the drugs, how our genetics affects that. And there are many different enzymes, mostly CYP enzymes in the liver, that are different depending on our genetics, and their metabolism is different depending on our genetics. So we have a panel, like many institutions have a panel, and many commercial companies have one, that we can run and see if the patient has a normal genotype or normal gene for that enzyme, or if it's abnormal in a way that would influence drug metabolism. The test we use is gene sequencing and not genotyping. The results are then implemented into the EMR. So when someone goes, let's say the patient is a non-responder to clopidogrel or Plavix, and then 5 years down the line they present with an MI, someone goes to starts typing clopidogrel in their EMR to prescribe it for them. Alert will come up in the EMR, kind of like this, to say, this patient has been genotyped. There's a so-called gene-drug interaction. That means that a patient will not respond to this drug. Here are a suggestion of alternative drugs like Prasugala, for example. And any questions, here's a number to call. And that number will go through to the pharmacogenetic pharmacist. This is what a part of a report looks like. So the red ones, there's a traffic light system. So anything in green is fine. Amber, use with caution. Use with caution, and then red, use with great caution. And use with great caution is typically drugs that are that have some sort of known indication or known interaction based on the genetics. And so we also have a mandatory education program for our pharmacists. So they, as soon as they come on staff, they're trained in this as well. Okay, so switch gears a little bit and talk a little bit about things that I do and some clinical case examples that we've seen and some nice success stories. So the first one is a case of a guy from Georgia, Dustin Bennett, who presented to us. He's a 22-year-old male, severe tremor, ataxic. He came from Waycross, Georgia. It's about an hour away from here. And he's seen multiple specialists, different hospitals, and no one's really figured out what's causing his symptoms. And it was his first visit to Mayo. And this is him. Let's see if we can get this playing. Yeah, okay. So you can see, it doesn't come up here, but you can see he's very ataxic, and he has a severe tremor. And this is what he's been like his whole life, really, and no one really can figure out what's going on. So unsurprisingly to this audience, we did whole exome sequencing, which he's never had before. And he has episodic ataxia type 1. Less than 100 people have been described with that condition. It's a mutation in a potassium voltage-gated channel, ion channel. And on reviewing the medical literature, people with this condition respond to acetazolamide and carbamazepine, which has never been tried for him. So we told the neurologist to try that. And that's him after treatment. So he's, by the way, he's still intellectually disabled, but we won't talk about that. You can see he's sitting, able to have a conversation. His walking is probably the most profound difference. So that's that one test with the information it led to greatly improved his quality of life. So another patient, so a 22-year-old female, hypotonia, basically from birth, muscle weakness, scoliosis, myopathy, joint hypermobility, short stature. She's been seen by many different people, misdiagnosed as or labeled as a metabolic myopathy or mitochondrial disease, and has actually had, so this is what she looks like. So you can see severe scoliosis and multiple orthopedic surgeries there to try and help. And she really just wanted to know what's wrong with her, or what's causing all her problems. So she actually had a research exome before, and which revealed one DNA, one pathogenic variant in RYR1. That was a couple of years ago. So I actually repeated her exome clinically and found a second hit. And so that's what she has. She has RYR1 related myopathy. And it was all consistent. Her clinical pathological findings in terms of her muscle biopsy showed multimonicore disease, which is all in keeping with this condition. So we actually identified that there was a trial going on at the NIH using N-acetylcysteine, which in zebrafish models has shown to be effective in reducing oxidative stress and rescued locomotor activity. So she's now enrolled in that trial and receiving N-acetylcysteine. And so that was nice. Of course, these were biallelic variants, so each parent was a carrier for this RYR1 mutations, and so we explained to them they're actually at risk for malignant hyperthermia. And so just a reminder that genetics as a family involves families, and implications for the child may have implications for parents in different ways. Another case, so this is a 37-year-old female, and she had a work-related injury mid-2015. She had basically hit her head. So she went to the ER, they had a brain MRI and CT. And so it revealed calcifications in the brain at the basal ganglia. And so after this is an unusual finding, and then after the imaging, she retrospectively reported she's had this resting tremor, recurrent headaches for the past 1 to 2 years, and it's present in her hands. And then her father reported that he's got some problems. His mother and so the patient's paternal grandmother also had problems as well. And so we looked at her and we looked at her other family members, and the phenotype really is resting tremor, headache, memory problems, basal ganglia calcification concerning for familial Farr's disease. And so the father had a CT, and he had basal ganglia calcifications. He had a mild tremor, forgetfulness, and also the patient's son, around 11 years old, had issues of recurring headache and tremor, which is unusual in an 11-year-old. So well, I'll tell you in a second. This is our CT, and so it shows basal ganglia calcifications. Basically, the white spots in the middle of the picture are the basal ganglia calcifications. And so we found a pathogenic variant in this gene that's been well described in causing familial Farr's disease, SLC20A2. And this I think this particular mutation has been reported once before. It's typically adult onset, so it was a little bit unusual that the child was presenting with problems at 11 years old. It is unfortunately a progressive disorder, it's caused by pathogenic variants in this solute carrier family 20 gene. This is the mutation that she has, and the other pathogenic mutations that have been described in the medical literature are there to see as well. So this is the family history. You can see it's a dominant inheritance pattern. It's a shame that our child is already affected at such a young age. So it doesn't appear to. She was in her 30s when she started getting symptoms. Her father was in his 50s, and the son is 11 years old. So there's no known history of anticipation or anything in this condition or in this gene. It's not a repeat disorder, but it was still unusual. Haven't really figured out why, or if it's purely because it's known about and we're looking at people earlier. All right, so I'll finish up with some case examples that my colleague, Dr. Elsia from Baylor, who I collaborate with, didn't talk about, and so I think this has kind of gone over already, but what is the rationale for metabolomic untargeted approach? Well, you're looking at small molecules, you're looking at actionable findings potentially, and people talk about a bridge between the genome and the phenotype. Current standards of testing involve a variety of targeted tests, which may or may not yield a diagnosis. And in the way that whole exome sequencing has become a very, very useful tool to diagnose people who have not had a diagnosis before, the thought is perhaps metabolomic profiling can play a similar role. And so really in the undifferentiated phenotypes such as intellectual disability or developmental delay, seizure, which there could be just so many different diagnoses. And the typical analytical pipeline is really using ultra high pressure liquid chromatography, tandem mass spec, with reference to a library, statistical analysis of analytes and compared to normal, and then interpretation of that based on analytes, their presence, their various abundance as well. So one kind of cool case that we had was a 4-year-old infant with developmental delay and hypotonia. So really very broad phenotype, could be one of many things, metabolic or not. He's had the usual litany of testing, very long chain fatty acids, lysosomal storage panel, MPS, CMA, metabolic screen. He's had CSF testing, so he's had a spinal tap. He's had a muscle biopsy, electron transport chain, so someone went down the kind of mitochondrial route with him, and also MRI brain. So very extensive, very expensive workup. So then he eventually had a exome and metabolomic profile. So the exome showed 2 VUSs in the DDC gene. So the DDC gene encodes for aromatic L-amino acid decarboxylase, which converts L-DOPA and 5-HTP to dopamine and serotonin respectively, which lead to the neurotransmitters homolinic acid and 5-hydroxyindolecetic acid respectively. And so when this is not working, there's a shunting to alternate pathway via COMT to produce 3-methoxytyrosine, or also known as 3-O-methyl-DOPA. And so the plasma metabolomic profile showed a marked elevation in 3-methoxytyrosine, a Z-score of 6.1, which is it's a pathognomic analyte for AADC. It can be seen with L-DOPA supplementation, of course, as well. So the child did have CSF neurotransmitters, which is appropriate to confirm the diagnosis, and it showed a very consistent and classic pattern for this condition, which is undetectable homovolinic and 5-hydroxyindolecetic acid and significantly elevated 3-methoxytyrosine. So that is this child's diagnosis. This is a nice slide. You can see the analytes that were detected and separated by their class. So amino acids, carbohydrates, cofactors, energy, lipid analytes, and nucleotide analytes. And hopefully it's clear that the analyte of interest is a large outlier, so it's very easy to see that there is something to hone in on worthy of further investigation. And this is just another representation of that. You can see the affected patient compared to unaffected patients. One or two of the higher up circles in the unaffected actually do have are getting supplementation with L-DOPA. This also is a way of functionally looking at some of the exome data. So you have these two VUSs that are very interesting, and it helps you think, well, this is more, perhaps more interesting because you're showing a biochemical pattern that fits with the VUSs. Okay, so another case is a 9-year-old male, very broad phenotype again, developmental delay, seizure, ataxia, normal microarray and standard metabolic workup. Exome showed two mutations in trans from two mutations in trans in this gene SLC13A5 that causes citrate transporter deficiency, and the plasma metabolomic profile showed a significantly elevated citrate in plasma. So you can see the Krebs cycle there. Red is showing a significant elevation. And we also did a CSF metabolomic profile on this patient that showed a quite decreased glutamine. So it's, there's a potential thought that glutamine, this SLC13A5, which is expressed in liver and brain, may also be a glutamate transporter as well. And that could be a possible mechanism behind the seizures and the intellectual disability. So we actually found 4 other cases, so 5 in total. And so you can see when you look at the 5 affecteds compared to the normals, there's a difference, and but when you normalize for other Krebs cycle intermediates, succinate, fumarate, and malate, there's an even slightly more obvious difference. And so it's not really been looked at in much detail because the technology is so new, but looking at different formulas and different algorithms to try and figure out these things. So that's kind of exciting in an area of further development. So that's really all I wanted to talk about. In terms of what we've talked about the advanced cancer, hereditary cancer, druggable gene targets, and the problems that are associated with that. Diagnostic odyssey, utilizing exome sequencing, as we're all doing, to find diagnosis and improve patient care. And then the kind of cool stuff with the metabolomic profiling, looking at biochemical pathway perturbations, with no available analyte testing, such as citrate transporter. You can't order plasma citrate, for example, and aiding in the interpretation of exome data. I think in future, down the line, we'll see more combining of different, omics platforms, genomics, metabolomics, transcriptomics, etc. Most are still in the research realm. And then the pharmacogenomics, gene-drug interactions, I think there's a lot of interest that is being gained in that area, and the FDA has guidelines on certain drugs, and that's only going to continue as well. So happy to take any questions and discussion.
Thanks. That's just amazing how these things all start to fit together in these big platforms. My question is about the use of z-scores. I'm used to a range, 50 to 100 is normal. Can you talk about your comfort level thinking about z-scores, and with this technology, do the z-scores, do the values in those z-scores change as you, maybe the levels of the analytes?
I completely see what you're saying, and my comfort level is it's a semi-quantitative test versus a quantitative, and I think that's the key, right? So I wouldn't call it a diagnostic test; it's a screening test. So if there's something there, it needs to be confirmed by the specific and targeted test. And so my comfort level is probably the same as yours, and looking at a z-score that can change based on the cohort you're using, and as that expands, that z-score could change as well. Yeah.
Thanks for your talk. That was very interesting. From a clinical genetics perspective, such as mine, I often wonder about when we get exome and genome data back, how much information is really in the database about any given gene, and I'm pretty certain that there's a lot of missing information in what we understand about functions of gene sequences and the implication of a sequence variant, for instance. And I wonder, so we're very good now at identifying sequence variants, and I'm wondering what you might suggest would be a process for us to fill in the other part, which would be potentially relates to the health record part of what you spoke about. But filling in those clinical, what's the process in our greater group for filling in the data related to gene information that's missing?
So yeah, I think that's an excellent question, and there's multiple different areas on the continuum that need to be filled in, right? So there's the sequencing, and I think someone alluded to it before, we're all using short read technology, which misses things, right? And so that's one step. Two is the bioinformatics. You give your exome to five different bioinformatations, you'll get 5 different sets of variants. So, for example, what we do when we get our let's say we get an exome back with no clear answer, we'll get the raw data back from the lab, the BAM file, and I have a buddy at the NIH that is a MD and bioinformatician that I send it to. We have a bioinformatics group that I send it to, and then there's another person, bioinformatician, so I send it to a couple of different people and see what they come up with. There's a lot of manpower, there's a lot of resources that are required for that. And I think, as well as you alluded to, there's no clear database that this is all being pumped into with, there are some databases, but incomplete. For example, you can look at XAG, but you don't get any clinical information with that. So, there's all sorts of issues with that. It's going to be a challenge. I don't know what the answer is, but I think we are improving, and we just need to keep doing what we're doing. Yeah. Okay, thank you.